All right, let's dive into pharmaceutical biotechnology. Think of this as a supercharged study session, especially for all you BSc Biotechnology Semester VI students out there gearing up for those exams.
Absolutely. We'll unpack those tough topics, tackle those questions you've sent in, and hopefully spark those aha moments that help everything really click.
We're going deep, folks, covering a whole semester's worth of material in this deep dive.
Let's kick things off with secondary metabolites. Those compounds produced by organisms that aren't directly involved in essential processes like growth and reproduction, think of them as a plant's secret weapon.
So, like a plant's way of saying back off,
Exactly. And we've learned to use these powerful compounds, especially in medicine.
which makes sense, but why are they called secondary metabolites in the first place? I mean, aren't all metabolites important for survival?
You're right, all metabolites have their roles, but the difference is between what's absolutely essential for basic survival versus what gives an organism that extra edge.
Okay, I'm following you.
Think of primary metabolites as the essential workers of a cell, involved in the daily grind of growth and reproduction. Your carbohydrates, proteins, lipids, nucleic acids keeping the cellular machinery humming.
Got it. So primary metabolites are the foundation, the essentials.
Exactly. Secondary metabolites come into play when an organism's basic needs are met and there are extra resources to spare. It's like investing in a security system or a fancy marketing campaign once your core business is running smoothly.
So they're like upgrades for a plant's defenses and interactions with the environment.
Precisely. These upgrades take many forms. Some secondary metabolites act as defenses against those pesky herbivores and pathogens. Others attract pollinators or help compete for resources.
The source material mentions this research goes way back to the early 20th century. Why did it take so long to recognize how important these compounds are?
Well, for one thing, secondary metabolites are often produced in smaller amounts than primary metabolites, making them harder to isolate and study. Plus, their chemical structures can be incredibly complex.
Like trying to find a needle in a haystack and then decipher his blueprint.
Exactly. We needed advanced techniques like chromatography and spectroscopy to really dive into their diversity and unravel those complex structures.
So the source material talks about secondary metabolites playing big roles in plant life, from defense to communication. Any specific examples?
Absolutely. Take alkaloids, a group of nitrogen-containing compounds that can be highly toxic to herbivores. One famous example is nicotine, produced by the tobacco plant.
So it's a natural insecticide protecting the plant from being eaten.
Exactly. Then there are volatile terpenes responsible for the scents of many flowers and fruits, acting as signals to attract pollinators.
It's like the plant is advertising its services with a fragrant perfume.
Precisely. And don't forget phenolic compounds, a diverse group that play multiple roles in defense, pigmentation, even communication, for example. Tannins found in tea, coffee, and red wine.
Ah, tannins. That dry, puckery feeling you get, so they're like a plant's built-in defense system.
Exactly. And these are just a few examples of the amazing diversity and complexity of secondary metabolites. We'll delve deeper into each group later, but first, let's talk about why they're so important for us humans.
Right. The source material mentions that they've been a real gold mine for drug discovery and development. Can you expand on that?
Absolutely. Humans have used plants for medicinal purposes for centuries, and many of these traditional remedies work because of the powerful secondary metabolites they contain. Modern medicine builds on this knowledge, isolating and synthesizing these compounds to create new drugs.
So we've been tapping into nature's pharmacy for a long time.
Exactly. And we're still discovering amazing compounds with potential therapeutic applications, like the anti-malarial drug artemisinin, derived from the sweet wormwood plant, which has saved countless lives, or the anti-cancer drug Taxol, isolated from the Pacific yew tree, which has revolutionized cancer treatment.
It's incredible how something a plant produces for its own defense can have such a huge impact on human health.
It really is. Now, let's get down to business and talk about the first major group you'll need to know for your exams: alkaloids.
Bring on the alkaloids. Time to dive into these potent nitrogen-containing compounds.
Let's start with the basics. Alkaloids are a diverse group of organic compounds that contain nitrogen atoms, usually within a ring structure, and they're produced by a wide range of organisms: plants, fungi, bacteria, even some animals.
So they're not just a plant thing.
Nope. Alkaloids are found throughout nature, playing a variety of roles, from defense to communication to even influencing the behavior of other organisms. But what's really fascinating from a pharmaceutical perspective is their strong biological activity.
And the source material mentions alkaloids have been a rich source of drugs. Any specific examples?
Absolutely. One of the most well-known is morphine, derived from the opium poppy. It's a powerful painkiller used for centuries to manage severe pain. It works by interacting with opioid receptors in the nervous system, blocking pain signals.
So a natural pain reliever targeting specific pathways.
Exactly. Another example is quinine. Historically extracted from the bark of the cinchona tree, a revolutionary treatment for malaria, interfering with the parasite's metabolic processes, effectively killing it off.
It's amazing how these plant-derived compounds can target specific mechanisms and pathogens to treat diseases.
It's the power of nature's pharmacy. But let's dig a little deeper into the structural diversity of alkaloids. You'll need to understand how they're classified based on their nitrogen-containing ring structures.
Right, the source material throws around terms like true alkaloids, protoalkaloids, and pseudoalkaloids. Can you break those down?
Sure. True alkaloids like morphine and quinine have their nitrogen atom incorporated into a heterocyclic ring, meaning the ring structure contains both carbon and nitrogen atoms. Protoalkaloids, on the other hand, have a nitrogen atom that's not part of a ring structure, like mescaline, a psychoactive alkaloid found in the peyote cactus.
So the key difference is whether the nitrogen atom is part of a ring or not.
Precisely. Now, pseudoalkaloids are a bit trickier. They may be derived from amino acids, but their nitrogen atom isn't part of a heterocyclic ring. Caffeine, the stimulant found in coffee and tea, is a classic example.
So even though they're all nitrogen-containing compounds, their structures lead to different classifications and potentially different biological activities.
Exactly. This structural diversity is why alkaloids are such a rich source of drugs. Now let's switch gears and talk about how these potent compounds are extracted and isolated from plants.
Okay, the source material mentions techniques like acid-base extraction and solvent extraction. How do those work?
All right, acid-base extraction uses the fact that most alkaloids are basic compounds, meaning they can accept a proton to form a positively charged ion, so you treat the plant material with an acidic solution, which converts the alkaloids into their salt form, making them soluble in water.
So you're essentially dissolving the alkaloids out.
Exactly. Then you use a nonpolar solvent, like chloroform or ether, to extract the alkaloids from that acidic solution, separating them from other water-soluble compounds. Finally, you treat the alkaloid-containing solvent with a base, converting the alkaloids back to their free base form, isolating them in their pure form.
It's a multi-step process, separating them based on their solubility in different solvents.
Precisely. Solvent extraction is another common technique. It uses a solvent that selectively dissolves the alkaloids from the plant material. The choice of solvent depends on the specific properties of the alkaloids you're targeting. Once dissolved, you can evaporate the solvent to isolate them.
So both techniques use selective solubility to separate the alkaloids from that complex mix in the plant material.
Exactly. These techniques have been instrumental in the discovery and development of many alkaloid-based drugs. Let's wrap up alkaloids by looking at some future directions in alkaloid research.
Right, the source material mentions multi-drug resistance and the potential of endophytic fungi. What's that all about?
Multi-drug resistance is a big problem in healthcare. It's when pathogens like bacteria and fungi evolve to resist the drugs we use to treat them, making infections much harder to treat, a major threat to public health.
It's like an arms race, us versus the pathogens.
Exactly. And we need new weapons to fight this resistance. Exploring new alkaloid structures, including those from endophytic fungi, is a promising avenue.
Endophytic fungi, what are those exactly?
Endophytic fungi live within plants without causing harm. They've co-evolved with their plant hosts over millions of years, producing a range of unique secondary metabolites, including alkaloids.
So like hidden allies producing potent compounds we can potentially use.
Exactly. And because they've evolved in such a specialized environment, they offer a rich source of novel alkaloid structures that could be effective against drug-resistant pathogens.
Like tapping into a secret arsenal.
Precisely. And that's just one exciting frontier in alkaloid research. Scientists are also using advanced computer modeling to predict their pharmacological properties and optimize their structures for drug development.
Combining the power of nature with the precision of technology.
Exactly. That's what makes this field so fascinating. So much potential for discovery and innovation, all driven by these incredible nitrogen-containing compounds, the alkaloids.
Well, I feel like I just got a crash course in alkaloid chemistry. Incredibly informative.
Glad to hear it. Now, we've talked about alkaloids, those potent nitrogen-containing compounds, but there's another massive group of secondary metabolites built from isoprene units. Let's dive into the world of terpenoids.
Okay, we've covered those nitrogen-packed alkaloids. Time to switch gears and explore a world built from isoprene units, terpenoids. I bet most people don't realize just how common these compounds are.
You're absolutely right. People encounter terpenoids all the time without even knowing it. That fresh pine scent in a forest. Terpenes. Vibrant flower colors attracting bees. Terpenoids again. The aroma of coffee. You guessed it. More terpenoids.
Wow, so they're not just some obscure scientific term, they're everywhere.
Exactly. Terpenoids, also known as isoprenoids, are a vast and diverse group of organic compounds built from those 5-carbon isoprene units. Think of isoprene as a molecular Lego block that nature uses to build an incredible array of complex structures.
It's amazing how much diversity nature can create from a simple building block. The source material mentions that terpenoids are classified based on the number of isoprene units they contain. Can you walk us through that classification?
Absolutely. The simplest are monoterpenes, made up of 2 isoprene units, giving them 10 carbon atoms. Many essential oils, like lavender and tea tree oil, are rich in monoterpenes. They're what give those oils their characteristic aromas.
So those essential oils I use for aromatherapy are basically concentrated doses of monoterpenes.
That's right. Now add another isoprene unit and you get sesquiterpenes. With 15 carbon atoms, these are often found in plants playing a role in defense against herbivores and pathogens. Artemisinin, that powerful antimalarial drug we discussed earlier, is a sesquiterpene lactone, a type of sesquiterpene with a ring structure.
So even within each class, there's a lot of structural variation.
Exactly. Now keep adding isoprene units and you get diterpenes, 20 carbons, triterpenes, 30 carbons, and tetraterpenes, 40 carbons. Diterpenes are found in resins and are responsible for that sticky sap you find on conifer trees. Triterpenes include important compounds like steroids, and cholesterol is actually built from a triterpene called squalene.
Wow, I never knew cholesterol was connected to terpenoids.
It's all connected. Finally, we have the tetraterpenes, which include carotenoids, the pigments that give fruits and vegetables their vibrant red, orange, and yellow colors.
So terpenoids are not just responsible for scents and flavors, but also for the amazing colors we see in nature.
Precisely. Let's dive into some specific examples of terpenoids that highlight their biological activities and their importance in medicine and industry.
The source material mentions artemisinin, taxol, limon, and squalene as prominent examples. Let's break those down one by one.
All right, let's start with artemisinin, as we mentioned. This sesquiterpene lactone, derived from the sweet wormwood plant, has been a game changer in treating malaria. It's incredibly effective. Even against drug-resistant strains of the parasite, its discovery was a major breakthrough, earning UU2, the Chinese scientist who isolated it, the Nobel Prize in Physiology or Medicine in 2015.
Talk about the power of nature's pharmacy.
Absolutely. Now let's move on to Taxol, a diterpenoid isolated from the bark of the Pacific yew tree. Taxol is a powerful anti-cancer drug used to treat a variety of cancers, including breast cancer, ovarian cancer, and lung cancer.
The source material mentions that Taxol works by stabilizing microtubules. Can you explain what those are and why that's important in cancer treatment?
Microtubules are like tiny scaffolds within our cells made of protein. They play a crucial role in cell division, helping to separate chromosomes during the process, ensuring that each daughter cell receives a complete set of genetic material.
So they're essential for cell division to happen correctly.
Exactly. Now, cancer cells are characterized by uncontrolled cell division. They divide rapidly and uncontrollably, forming tumors and spreading throughout the body.
So if you can disrupt that process of cell division, you can potentially stop cancer.
That's the idea. And that's where Taxol comes in. It binds to microtubules and stabilizes them, preventing them from breaking down. This disrupts cell division, effectively stopping those cancer cells from multiplying.
So it's like throwing a wrench into the machinery of cell division, specifically targeting those rapidly dividing cancer cells.
Exactly. Let's move on to limon, a monoterpene that gives citrus fruits their characteristic aroma. You'll find it in oranges, lemons, limes, and grapefruits.
I love that fresh citrus scent. It's so uplifting.
And not only does lemon smell great, but it might also have some health benefits. It's been shown to have anti-inflammatory and antioxidant properties, and it's even being investigated for its potential as an anti-cancer agent.
So adding that squeeze of lemon to my water might be doing more than just adding flavor.
Exactly. Now let's talk about squalene, a triterpene that's a precursor to cholesterol and other steroid hormones in our bodies. You might be surprised to learn that squalene is also found in high concentrations in shark liver oil.
Shark liver oil. That sounds a bit intense.
It is. Squalene is a valuable compound, and shark liver oil has been used for centuries in traditional medicine for its purported health benefits. However, Harvesting sharks for their liver oil has raised ethical concerns about the sustainability of shark populations.
It's a reminder that we need to be mindful of the ethical implications of using natural resources, especially when it comes to endangered species.
Absolutely. Now let's shift our focus to the biosynthesis of terpenoids. You'll need to understand the two main pathways involved in their production: the mevalonate pathway and the mesylerythritol phosphate, MEP pathway.
The source material mentions these pathways, but I'm a bit lost on the details. Can you give us a simplified explanation?
Think of these pathways as two different assembly lines inside the cell, each using different starting materials and enzymes to produce isoprene units, those molecular Lego blocks we talked about. The mevalonate pathway starts with acetyl-CoA, a common molecule involved in many metabolic processes, and converts it through a series of steps into isopentenyl pyrophosphate, IPP, and dimethylylly pyrophosphate, DMEPP, two activated forms of isoprene.
So it's a multi-step process, each step catalyzed by a specific enzyme to create those essential building blocks.
Exactly. The MEP pathway, also known as the non-mevalonate pathway, starts with pyruvate and glyceraldehyde 3-phosphate, two other common metabolic intermediates. It uses a different set of enzymes to convert these starting materials into IBP and DMEPP.
So two different pathways, but both leading to the production of those crucial isoprene units.
Precisely. And these pathways operate in different cellular compartments. The mevalonate pathway is primarily active in the cytosol, the fluid-filled space inside the cell, while the MEP pathway operates in plastids, specialized organelles found in plant cells.
It's amazing how the cell has these compartmentalized pathways, each contributing to the overall production of these essential molecules.
It really shows the complexity and efficiency of cellular processes. Now, let's wrap up terpenoids by looking at some exciting research directions in this field.
Right, the source material mentions exploring plant-derived terpenoids as potential leads for new drugs and using synthetic biology approaches to enhance terpenoid production. Can you elaborate on those?
Absolutely. As we've seen, plant-derived terpenoids have already given us some remarkable drugs, but there's still so much potential waiting to be unlocked. Researchers are constantly screening plants for new terpenoids with potential therapeutic applications, especially in the fight against drought-resistant pathogens and cancer.
So it's a continuous search for new weapons in nature's arsenal.
Exactly. And synthetic biology is playing an increasingly important role in this search. Scientists are using genetic engineering techniques to manipulate metabolic pathways in microorganisms and plant cells, essentially turning them into tiny factories for producing specific terpenoids.
It's like we're taking inspiration from nature and using technology to create custom-designed terpenoid producers.
Precisely. And this has huge implications for the pharmaceutical industry, potentially making it possible to produce valuable terpenoids in large quantities and at lower costs.
That's incredibly exciting. It opens up so many possibilities for drug development and other applications.
It really does. Now, with terpenoids thoroughly explored, let's shift our focus to another powerhouse group, the phenolic compounds. Get ready for a deep dive into the world of antioxidants, pigments, and more.
Okay, ready to dive into that last group of secondary metabolites, those phenolic compounds.
Phenolic compounds are incredibly versatile. They're responsible for those vibrant colors and many fruits and flowers. They protect plants from damage, and they even have those potent antioxidant properties that can be good for human health.
Sounds like they're real multitaskers.
Definitely. What makes them unique is that hydroxyl group attached to an aromatic ring. That ring-shaped structure made of carbon atoms. This simple feature leads to a huge diversity of phenolic compounds, each with their own properties and functions.
Right, the source material mentions several classes: flavonoids, phenolic acids, tannins, and lignans. I want to break those down one by one, maybe starting with those flavonoids.
Flavonoids are probably the most well-known. They're responsible for much of the color we see in plants, especially reds, blues, and purples in those fruits and flowers. Think blueberries, red grapes, those beautiful purple irises.
They're like the plant world's artists, painting nature with vibrant colors.
Exactly. But flavonoids are more than just pigments. They have roles in plant defense, protection against UV radiation, and even attracting pollinators. And for us, they're a valuable source of antioxidants.
Antioxidants, those are important for our health, right?
Our cells are constantly under attack from free radicals, those unstable molecules that can damage DNA, proteins, cell membranes. This damage is called oxidative stress, and it contributes to aging and a lot of diseases: heart disease, cancer, Alzheimer's disease.
So it's like a constant battle, our cells versus those harmful free radicals.
Exactly. Antioxidants are like the cellular defense squad. They neutralize those free radicals, prevent damage, and protect ourselves from oxidative stress.
So the more antioxidants we get, the better equipped our cells are to fight back.
That's the idea. Flavonoids are particularly potent antioxidants, and they're abundant in fruits, vegetables, and things like tea and red wine.
Eating those colorful fruits and veggies is like giving ourselves a boost of protection then.
Exactly. Now let's look at some specific flavonoids. One well-known one is quercetin, found in onions, apples, berries, and grapes. Quercetin has anti-inflammatory, antiviral, even anti-cancer properties.
Wow, a multi-talented flavonoid.
That's right. Another important group are the anthocyanins, responsible for those red, blue, and purple colors in many fruits and veggies. They're in blueberries, strawberries, raspberries, blackberries, even red cabbage.
So those colorful fruits and veggies are not just pretty to look at, but they're packed with beneficial flavonoids too.
Exactly. Anthocyanins are powerful antioxidants linked to improved heart health, protection against cognitive decline, even a reduced risk of certain cancers.
It's like nature's giving us a delicious and beautiful way to stay healthy.
It does seem that way. Now let's move on to phenolic acids, another important group in those phenolic compounds. Phenolic acids are found in a variety of plant-based foods and drinks, including coffee, tea, fruits, vegetables, and whole grains.
The source material mentions caffeic acid and ferulic acid as examples. Can you tell us more about those?
Caffeic acid, as its name suggests, is found in coffee beans, a potent antioxidant with anti-inflammatory properties. Ferulic acid is another important one, found in whole grains, fruits, and vegetables. It's a powerful antioxidant shown to protect against UV damage and even have anti-cancer effects.
So both caffeic acid and ferulic acid are like little shields protecting our cells.
Exactly. Now let's move on to tannins. Those are known for their astringent properties. You've probably experienced that astringent effect if you've ever had red wine or unripe fruit, that dry, puckering sensation.
I know that feeling. What causes that?
Tannins have a complex structure that allows them to bind to proteins. When you have red wine or unripe fruit, the tannins bind to proteins in your saliva, creating that astringent feeling.
So it's a chemical interaction between tannins and proteins that creates that unique sensation.
Exactly. Tannins are also important in plant defense. They can deter herbivores, and they can protect against pathogens.
Like the plant security system.
That's right. And tannins may also have health benefits for us. They've been shown to have antioxidant and antimicrobial properties, and some are even investigating their potential role in preventing certain cancers.
Wow. So those puckering compounds can also have beneficial effects.
It really shows the complexity and diversity of the plant world. Okay, let's wrap up phenolic compounds by talking about lignins.
Lignins, those sound familiar. Are they something to do with wood?
You got it. Lignins are complex polymers that make up a significant portion of plant cell walls, providing structural support and rigidity. They're essentially the wood in woody plants, also found in the fibrous parts of fruits and vegetables.
So they're what give plants their strength and structure.
Exactly. Lignins also play a role in plant defense, making it harder for pathogens to get into plant tissues.
So like the plant's armor.
That's a great way to put it.
Yeah.
And lignins might also have health benefits. They've been shown to have antioxidant properties. And there's research looking into their potential role in lowering cholesterol levels and reducing the risk of certain cancers.
Incredible how something that gives plants structure can also have positive effects on our health.
It is remarkable. We've covered a lot in this deep dive into the world of secondary metabolites, their diversity, their roles in plants, their amazing applications in medicine and other industries, and their potential benefits for us.
It's been a fascinating journey exploring these often overlooked but incredibly important compounds.
I hope you've gained a new appreciation for their complexity and wonder and how important they are in our world.
Always remember to keep exploring, questioning, and learning. The world of secondary metabolites is vast and always changing, with so much still to discover.
And every time we dive deeper into science, it's a chance to find something new and amazing.
Thanks for joining us on this deep dive into the world of secondary metabolites. Keep exploring, and best of luck with those exams.